So the final step in constructing a recombinant DNA molecule is to join together the vector molecule and the DNA we want to clone. This crucial process is called ligation. It involves an enzyme known as DNA ligase, which catalyzes the joining of these DNA fragments. Now let's talk about how DNA ligase actually works. All living cells naturally produce DNA ligases. They're essential for DNA repair and replication. However, in genetic engineering, we usually use a purified form of this enzyme, often derived from E. coli bacteria that have been infected with T4 phage. Within the cell, DNA ligase performs a vital function. It repairs any discontinuities or breaks that might occur in the strands of double-stranded DNA. This repair activity is what makes it so useful in cloning, as it seals nicks in the backbone of DNA molecules, ensuring they are intact and stable. Understanding the mode of action of DNA ligase helps us appreciate its role in cloning strategies. Without this enzyme, joining DNA fragments into a stable recombinant molecule would be nearly impossible, which is why it's such a cornerstone of genetic engineering techniques. Ligases are fascinating enzymes because they play a crucial role in joining nucleic acid molecules together. These molecules can be either DNA or RNA, which is why we have specific names for the enzymes: DNA ligase and RNA ligase. When we talk about DNA ligase, we're referring to its ability to catalyze the formation of a phosphodiester bond. This bond forms between the 5-phosphate group of one nucleotide and the 3-hydroxyl group of another, effectively sealing the DNA backbone. In nature, the primary function of DNA ligase is to repair single-strand breaks, or discontinuities, that can happen during DNA replication or recombination. Think of it as a kind of molecular glue that helps maintain the integrity of the genetic information. In the context of recombinant DNA technology, ligases are equally important. They enable us to join a piece of DNA of interest, often called an insert, with a vector molecule. This process is what we refer to as ligation. It's a fundamental step in cloning strategies, allowing us to manipulate DNA in precise ways for research or therapeutic purposes. Cloning strategies are essential techniques we use to insert a foreign DNA fragment into a vector, like a plasmid, so it can replicate inside a host cell. The success of cloning really depends on how well we prepare and join the ends of both the DNA fragment and the vector. There are several major strategies we can use. One is blunt end ligation, where the DNA ends are flush and can be directly joined. Then there's cohesive or sticky end ligation, which takes advantage of overhangs that can pair up specifically. We also have cloning using linkers, which are short, double-stranded DNA sequences that help create compatible ends. Similarly, adapters are used to attach to DNA fragments to facilitate cloning. Lastly, homopolymer tailing involves adding a series of identical nucleotides to the ends of DNA to assist in cloning. Each of these methods differs based on the nature of the DNA ends and how the ends are joined together during the ligation process. Understanding these strategies helps us choose the most efficient method for our specific cloning needs. Blunt ends are DNA ends where both strands end at the same nucleotide position, meaning there are no overhangs. These kinds of ends are typically generated by restriction enzymes like SMI or by DNA polymerases that either fill in or chew back overhangs. Now let's talk about the characteristics of blunt ends. The key thing to remember is that there are no single-stranded overhangs present. This means the ends don't have any extra bases sticking out. Also, because there are no overhangs, these ends don't have complementary base pairing. And importantly, ligation, the process of joining DNA fragments, doesn't depend on hydrogen bonds between overhangs, since they simply aren't there. Moving on to how blunt end ligation actually works. Both the vector and the insert DNA are cut to produce blunt ends, so they can fit together. The enzyme responsible for sealing these ends is DNA ligase, commonly T4 DNA ligase. It catalyzes the formation of a phosphodiester bond between the 3O group of one end and the 5-phosphate group of the other. To do this, the ligase needs ATP, which acts as a cofactor to power the reaction. Because there are no base pairs stabilizing the ends, blunt-end ligation tends to be less efficient than sticky-end ligation. That's an important point to keep in mind, especially when planning cloning strategies. In summary, blunt-end ligation involves joining DNA fragments with no overhangs, relying solely on the enzyme's activity to create bonds, and generally results in lower efficiency compared to sticky-end ligation. Let's talk about the advantages of blunt-end ligation. One key benefit is that any blunt-ended fragment can be ligated to any other blunt-ended DNA fragment. This makes the process quite flexible because you don't need to worry about matching restriction sites. In other words, you can join any two blunt ends without the need for compatible overhangs, which simplifies some cloning procedures. Now, moving on to the disadvantages. One major issue is that the ligation efficiency tends to be lower compared to sticky ends. Blunt ends don't have overhangs to guide the ligation process, so the chances of successful joining are reduced. Additionally, there's a higher risk of the vector self-ligating, meaning the vector could close up on itself without incorporating the insert. This can make cloning less efficient and require extra steps to identify the correct constructs. Another important point is that blunt-end ligation doesn't allow for directional cloning. Without sticky ends, you can't control the orientation of the inserted fragment, which can be a limitation depending on your experimental goals. Overall, while blunt-end ligation offers some straightforward advantages, like not needing compatible restriction sites, it also comes with notable challenges that you should keep in mind during your cloning strategies. Cohesive ends are produced when restriction enzymes cut DNA in a staggered manner, which creates short single-stranded overhangs. These overhangs are important because they can form hydrogen bonds with complementary sequences, making them very useful in recombinant DNA technology. Examples of enzymes that generate such ends include E-CoRI and HindII, which are commonly used in labs. Now, let's talk about the characteristics of these cohesive ends. As you can see, they contain these single-stranded overhangs, and because of their specific sequences, they can pair with matching overhangs on other DNA molecules. This pairing through hydrogen bonds gives a high level of specificity during ligation, meaning the pieces are more likely to connect correctly. Moving on to the mechanism of sticky-end ligation, the process begins when both the vector and the insert are cut with the same restriction enzyme. This step produces complementary overhangs that can base pair with each other. These hydrogen bonds temporarily hold the DNA fragments in place, which makes the subsequent ligation step much more efficient. Finally, DNA ligase comes into play. It seals the sugar phosphate backbone by forming phosphodiester bonds, effectively gluing the DNA pieces together. Those initial hydrogen bonds are crucial because they stabilize the fragments during ligation, increasing the overall efficiency of the process significantly. This is why sticky-end ligation is such a powerful tool in genetic engineering and cloning. Now, let's take a closer look at the advantages of sticky-end ligation. First, it offers a higher ligation efficiency compared to blunt ends, which means it's generally more successful and reliable when joining DNA fragments. This is because the sticky ends can form specific, stable base pairs, making the ligation process more precise and efficient. Additionally, when using two different enzymes, sticky ends allow for directional cloning. This is a huge benefit because it helps us insert DNA fragments in a specific orientation, which is crucial for many applications. However, there are some limitations to keep in mind. Sticky-end ligation requires compatible restriction sites, so you need to have the right enzymes that produce compatible overhangs. This can sometimes be restrictive because you're limited by the availability of suitable recognition sequences in your DNA. Overall, sticky ends are a powerful tool in molecular cloning because of their efficiency and specificity, but it's important to consider these limitations when planning your experiments. Joining of two blunt-ended fragments in a test tube is not very efficient because the ligase enzyme struggles to catch hold of the molecule to be ligated. Essentially, the enzyme has to wait for chance associations to bring the ends together, which makes blunt-end ligation less reliable. To improve this process, we typically perform blunt-end ligation at high DNA concentrations, increasing the chances that the ends will come into contact. In contrast, ligation of complementary sticky ends is much more efficient. That's because these sticky ends can base pair with each other through hydrogen bonding, creating a relatively stable structure for the ligase enzyme to work on. However, if the phosphodiester bonds aren't formed quickly enough, these sticky ends can fall apart again before the bonds are sealed. So, why is sticky end ligation better? The key reason is that the base pairing provides a kind of temporary anchor, making it easier for the enzyme to complete the ligation. This stability leads to higher success rates, which is why sticky end ligation is generally preferred in molecular cloning and genetic engineering. This concept underscores the importance of DNA structure and enzyme activity in efficient DNA manipulation, fitting into our overall theme of understanding how enzymes facilitate genetic modifications. When it comes to cloning, the sticky ends are really important because they enable the DNA fragments to pair up with the vector DNA. These sticky ends can be generated by digesting both the vector and the DNA to be cloned with the same restriction endonuclease. Alternatively, you can use different enzymes that produce compatible sticky ends, which can also work well. However, it's not always possible to do this, especially when the restriction sites don't match up or aren't available. So, a common situation we encounter is when the vector molecule has sticky ends, but the DNA fragments we want to clone are blunt-ended. This mismatch can make cloning more challenging because blunt ends don't naturally pair like sticky ends do. To overcome this, there are a few methods we can use. One approach is to add linkers, which are short, double-stranded DNA sequences that contain the desired sticky ends. Another method involves adapters, which are similar but can be more versatile in attaching different sequences. And there's also the option of producing sticky ends by homopolymer tailing, where we add a string of identical nucleotides to the blunt ends to create sticky ends. Now, why is sticky end ligation considered better? Well, because sticky ends tend to pair more specifically and efficiently, making the ligation process more successful. They help ensure that the right DNA fragments are joined in the correct orientation, which is crucial for downstream applications. So, while blunt-end cloning is possible, sticky end ligation generally provides a more reliable and efficient method for cloning DNA fragments. Linkers are short, synthetic, double-stranded DNA fragments that contain a restriction enzyme recognition site. They are used to convert blunt ends into cohesive, or sticky, ends, which makes the subsequent ligation process much more efficient. Typically, these oligonucleotide sequences are around 8 to 12 base pairs long and include a specific restriction site within their sequence. One key point to remember is that they have blunt ends, which allows them to be ligated to target DNA molecules that also have blunt ends. For example, in this case, the linker contains a BAM-HI restriction site. Even though the linker itself is blunt-ended, it can be attached to DNA molecules that have been cut with BAM-HI, which produces sticky ends. DNA ligase plays a crucial role here, as it can attach these linkers to the ends of larger blunt-ended DNA molecules. Because the linkers are present in very high concentrations during the reaction, this process becomes highly efficient. This high concentration drives the ligation reaction forward, making it a very effective method to prepare DNA for cloning. Overall, the use of linkers simplifies the process of cloning by providing a way to introduce specific restriction sites onto blunt-ended DNA, facilitating subsequent steps like digestion and ligation in the cloning workflow. Many linkers will attach to each end of the double-stranded DNA molecule. When we digest these with BAM-HI, it cleaves the linker chains at their specific sequences, which results in a large number of cleaved linkers and a DNA fragment with BAM-HI sticky ends. This modified DNA fragment is now ready to be ligated into a cloning vector that has been cut with BAM-HI as well. Now let's look at the mechanism. A blunt-ended DNA fragment can be ligated with linkers using DNA ligase. After this ligation, the linker ligated DNA is treated with the same restriction enzyme that recognizes the linker site. This digestion creates cohesive, or sticky, ends on the insert. Finally, the insert, now with sticky ends, can be ligated into a vector that was cut with the same restriction enzyme. This process, known as cloning using linkers, allows us to insert DNA fragments with greater flexibility and precision. Let's talk about some advantages of using linkers in cloning. First, they enable us to insert blunt-ended DNA into vectors that have sticky ends. This is a significant benefit because it increases our cloning efficiency, making the process smoother and more effective. However, there are also some limitations we need to keep in mind. For example, if the DNA insert contains internal recognition sites for the same enzyme we're using, unwanted digestion can occur. This can complicate the process and reduce yield. Another important point is that when using linkers, if the blunt-ended DNA we want to clone has one or more BAM high recognition sequences, there's a drawback. Specifically, the restriction step that we use to cleave the linkers and produce sticky ends might also cut into our blunt-ended DNA molecule itself. This can lead to unwanted cleavage and affect our cloning efficiency. So, while linkers are a useful tool, understanding their limitations helps us plan better and avoid potential pitfalls in our cloning strategies. Adapters are short, synthetic, double-stranded DNA molecules that contain one blunt end and one cohesive end. They are used to convert blunt-ended DNA into sticky-ended DNA without needing restriction digestion after ligation. One end of the adapter is blunt, which allows it to ligate easily to the insert DNA. The other end has a preformed sticky overhang, which is key for facilitating the insertion into a vector with complementary overhangs. The mechanism works by ligating the adapter to the blunt-ended DNA. Once attached, the cohesive end of the adapter can insert into a vector that has matching overhangs, making the cloning process more efficient. Unlike linkers, adapters already contain sticky ends, so there's no need for additional digestion after ligation. This approach simplifies cloning workflows, especially when you want to generate sticky ends from blunt DNA fragments, making the overall process more straightforward and reliable. So, in practice, the sticky ends of individual adapter molecules could actually pair with each other, forming dimers. This means that even if we design them to be sticky, they might end up creating new blunt-ended DNA molecules instead. While it's possible to recreate these sticky ends by cutting with a restriction endonuclease, that approach would defeat the purpose of using adapters in the first place, since we're trying to avoid additional digestion steps. The key to solving this problem lies in the chemical structure of the adapter ends themselves. Normally, the two ends of a polynucleotide strand are chemically distinct. The 5' carries a phosphate group, often called the 5' and the 3' has a hydroxyl group, or 3O. In double-stranded DNA, the two strands are oriented anti-parallel, meaning one runs from 5 to 3 in one direction, and the other runs in the opposite direction. So, each end of a double-stranded DNA molecule has one 5' terminus and one 3O terminus. Ligation, or the joining of DNA fragments, only occurs between these specific ends, the 5' of one strand and the 3O of another. This structural characteristic is crucial because it ensures that ligation happens in a controlled, specific way, which is the foundation for successful cloning using adapters. To overcome the problem of self-ligation among adapters, scientists have designed specially synthesized adapter molecules. These adapters are made so that their blunt end is identical to natural DNA, which allows them to attach to DNA fragments. However, their sticky end is different, and this difference is crucial. The key modification is on the 5' terminus, which normally has a phosphate group, but in these adapters, it's missing and replaced with a 5O group. This means the adapter's sticky end has a 3O terminus that remains the same, but the 5' is modified to prevent unwanted ligation. Now, DNA ligase, the enzyme responsible for joining DNA strands, can't form a phosphodiester bond between a 5O and a 3O. This modification ensures that adapters can be ligated to blunted DNA fragments, but they won't ligate to themselves, preventing the formation of adapter dimers. This is a clever way to facilitate cloning using adapters, making the process more efficient and specific. After the attachment of adapters, the abnormal 5' terminus is converted to the natural 5'. This step is crucial because it prepares the DNA fragment for further processing. When we treat it with the enzyme polynucleotide kinase, it adds a phosphate group to the 5' end, creating a 5'. This phosphorylation results in a sticky-ended fragment, which means it has overhanging bases that can easily pair with complementary sequences. These sticky ends are essential for efficient insertion into an appropriate vector. Cloning using adapters thus involves attaching these specially designed sequences to the DNA fragments, making them compatible with vectors, and facilitating their incorporation into the cloning process. This method streamlines the cloning workflow and enhances the precision of gene insertion. Now, let's take a look at the advantages of using adapters in cloning. One of the key benefits is that they are more efficient than traditional linkers. This means we can save time and reduce the complexity of our cloning process. Additionally, using adapters helps us avoid an extra restriction digestion step, which can often be a bottleneck or add to the overall time and effort required. However, it's important to be aware of the limitations too. For example, adapters may self-ligate if they are not properly treated, which can lead to unwanted byproducts or background noise in our experiments. When cloning using adapters, careful handling and treatment are essential to prevent this issue. Overall, adapters can be a powerful tool, but they require some attention to detail to ensure successful cloning. This ties into our broader discussion on optimizing cloning strategies for efficiency and reliability. Homopolymer tailing can also produce sticky ends on a blunted DNA molecule. Now, a homopolymer is simply a polymer where all the subunits are the same. For example, a DNA strand made up entirely of deoxyguanosine nucleotides would be considered a homopolymer. Tailing involves using an enzyme called terminal deoxynucleotidyl transferase. This enzyme adds nucleotides onto the 3-hydroxyl, or 3-OH, end of a double-stranded DNA molecule. What's interesting here is that by adding a homopolymer tail, even on a blunted DNA, we can create sticky ends. These sticky ends are useful because they can facilitate specific and efficient ligation with complementary sequences, which is a key aspect in cloning and genetic engineering. So, in the context of our overall theme, understanding homopolymer tailing gives us a powerful tool to modify DNA ends in a controlled way, opening up many possibilities for manipulating DNA molecules in the lab. When we carry out this reaction in the presence of just a single deoxyribonucleotide, it results in the formation of a homopolymer tail. This means that only one type of nucleotide is added repeatedly, creating a stretch of identical bases. Now, for successful ligation, these homopolymer tails need to be complementary to the vector DNA. Specifically, if we have a poly-C tail, it must pair with a poly-G tail on the vector. This complementary base pairing is crucial because it ensures that the DNA molecules can hybridize correctly. When we mix the DNA molecules, these homopolymer tails align through base pairing, facilitating the ligation process. So, in essence, homopolymer tailing is a key step that allows us to prepare DNA fragments for efficient and specific cloning by ensuring they can properly anneal and ligate with their complementary vectors. In practice, the poly-G and poly-C tails we add are rarely of exactly the same length. This means that the recombinant molecules we produce often have nicks and discontinuities in their DNA strands. To fix these issues, we perform a two-step repair process. First, we use clenopolymerase to fill in the gaps or nicks in the DNA. Then, DNA ligase comes in to seal the final phosphodiester bonds, ensuring the DNA strands are continuous and stable. Once these recombinant molecules enter the host cell, the cell's own DNA polymerase and DNA ligase take over. They complete the repair process, finalizing the construction that we initially started in the test tube. This process of adding homopolymer tails is a crucial step in ensuring the integrity and functionality of the recombinant DNA. Let's take a look at the mechanism behind homopolymer tailing. Essentially, terminal transferase is an enzyme that adds a series of identical nucleotides, such as a poly-DA tail, to the three ends of the insert DNA. This process creates a single-stranded tail that can be used for cloning purposes. Next, the vector DNA is treated to receive complementary tails, for example, a poly-DT tail. When these tails come into contact, they anneal through base pairing, which helps bring the insert and vector together. Finally, DNA ligase acts to seal any nicks in the sugar phosphate backbone, resulting in a stable recombinant molecule. Now, moving on to the advantages. This method doesn't require restriction enzymes, which makes it quite straightforward and useful, especially when you don't have suitable restriction sites available in your DNA. However, there are some disadvantages to keep in mind. This method is less specific, meaning it can sometimes lead to nonspecific ligation events. It's also not ideal for directional cloning because it doesn't provide control over the orientation of the insert. In fact, due to these limitations, homopolymer tailing has been largely replaced by more modern, precise cloning techniques. Overall, homopolymer tailing was an important step in the evolution of cloning methods, but today, we rely on more advanced tools for efficiency and accuracy. All right, let's take a closer look at the different cloning strategies outlined in this table. First, we have blunt-end cloning, which involves no overhangs. This method is generally low in efficiency because it relies on ligating two blunt ends, which can be less precise and less efficient. It typically requires just ligase to seal the ends together. Next, we have sticky-end cloning, which uses overhangs. These overhangs are single-stranded regions that can base pair specifically, making this method highly efficient. Because of that, it's often preferred when high efficiency is needed. For this, we use restriction enzymes to generate the overhangs, followed by ligase to seal the nicks. Then there are linkers. These are short sequences that can convert blunt ends into sticky ends. They're quite versatile because they allow us to take a blunt-ended fragment and make it compatible with sticky-end cloning, thus increasing efficiency. The process involves ligase and restriction enzymes, making it a flexible option. Moving on, adapters are pre-made sticky ends that can be ligated to blunt-ended fragments. This approach also offers high efficiency and is useful when working with fragments that don't naturally have sticky ends. Again, ligase is the key enzyme here. Finally, we have homopolymer tailing, which involves adding polynucleotide tails to DNA fragments. This method provides moderate efficiency and doesn't require restriction enzymes. Instead, we use terminal deoxynucleotidyl transferase, or TDT, along with ligase if needed, to add these tails. Overall, these strategies give us a toolkit of options, each suited to different needs depending on the fragment type and desired efficiency. It's all about choosing the right approach for the specific cloning project. These complementary bases, meaning the T and A overhangs, allow the PCR product to anneal to the vector. Once they are paired, DNA ligase comes in and seals the backbone, which results in a stable recombinant plasmid. This method is particularly useful because it allows us to clone PCR products directly without the need for restriction digestion, making the process faster and simpler. Now, one popular technique that uses this principle is TA cloning, or thymine, adenine cloning. It's a straightforward and widely used method in molecular biology. The key idea here is that certain DNA polymerases naturally add an extra adenine nucleotide at the 3' end of PCR products. This adenine can then pair easily with thymine overhangs that are present in specially designed vectors. TA cloning is a restriction enzyme, independent method, meaning we don't need to cut the DNA with enzymes beforehand. Instead, we insert the PCR product directly into the plasmid vector. The name TA comes from the fact that it relies on the complementary base pairing between thymine overhangs on the vector and adenine overhangs on the PCR product. This simple pairing makes cloning more straightforward and accessible, especially when speed is important. Now let's take a closer look at the vector end in TA cloning. On the 3 end, we have a thymine, or T, and on the 5 end, an adenine, or A. This creates a specific overhang that is crucial for the cloning process. Moving to the vectors with T overhangs, these vectors are specially engineered to contain single thymine residues at their 3 ends. This is important because these T residues are complementary to the overhangs that we find on our PCR products. Picture it this way. The 5 end of our DNA fragment has an A, and the 3 end also has an A, which pairs with the T on the vector. This complementary base pairing allows for a very specific and efficient joining of the PCR product to the vector. Now, the principle behind TA cloning hinges on 3 key molecular properties. The first is the addition of A overhangs by an enzyme called Taq polymerase. Many thermostable DNA polymerases, especially Taq, have what's called terminal transferase activity. This means that after they synthesize DNA during PCR, they tend to add a single adenine nucleotide, an A, at the 3 ends of the PCR product. Think of it as the polymerase leaving a little sticky end of A's on our PCR product. This is what makes TA cloning possible. These overhangs can readily pair with the T overhangs on our vector, facilitating the cloning process. So, in summary, the key to TA cloning is this A overhang addition by Taq polymerase, enabling the PCR product to seamlessly ligate with the T overhang vectors, making cloning straightforward and efficient. Now let's look at the ligation step. Here, the PCR fragment anneals to the vector through a T-base pairing. This pairing is crucial because it allows the fragment to align correctly with the vector. Once aligned, DNA ligase comes into play. Its role is to form phosphodiester bonds, effectively sealing the DNA backbone and creating a continuous, stable molecule. The end result of this process is a recombinant plasmid that contains our PCR insert. This is a key step in TA cloning because it ensures that our insert is correctly incorporated into the vector, setting the stage for the next steps in cloning and expression. All right, let's walk through the steps involved in TA cloning. First, we have PCR amplification. Here, the DNA fragment that we want to clone is amplified using PCR. It's important to note that we use Taq DNA polymerase for this step because it adds those crucial A overhangs at the 3 ends of the PCR products. We want to avoid proofreading polymerases like Fu because they don't add these A overhangs, which are essential for the cloning process. The result of this step is a PCR product that has those 3 adenine overhangs, making it compatible for the next step. Now, moving on to the preparation of the TA vector. The cloning vector is first linearized and then modified to contain 3 thymine overhangs, which are complementary to the A overhangs on our PCR product. Common vectors used for TA cloning include PGEMT-EZ, PCR21, and PTZ57RT. These vectors are designed to facilitate easy cloning because they also carry other important features. For example, they contain antibiotic resistance genes, so we can select for successful transformants, multiple cloning sites for inserting our DNA fragment, and a blue-white screening system to help identify colonies with the inserted DNA. So to sum up, in TA cloning, we're leveraging the natural A overhangs added by Taq polymerase to efficiently insert our PCR product into a specially prepared vector that has complementary T overhangs. This method is simple and effective, especially for cloning PCR products directly. Now let's move on to step 3, the ligation reaction. At this stage, we combine our PCR product, which contains the gene of interest, with the TA vector and ligase enzyme. This mixture includes the TA vector itself, the PCR insert, ligase buffer to maintain the right conditions, and T4DNA ligase, which is the enzyme responsible for sealing the nicks and joining the DNA fragments together. The goal here is to create a recombinant plasmid that carries our gene of interest within the vector. Once the ligation is complete, we move to step 4, transformation. This is where we introduce our recombinant plasmid into competent bacterial cells, most often Escherichia coli, to allow for amplification. There are a couple of common methods to do this. Heat shock, where we briefly expose the cells to a high temperature to make their membranes more permeable, or electroporation, which uses an electric pulse to facilitate DNA entry. After the bacteria take up the plasmid, we plate them on media containing antibiotics. This step is crucial because only the bacteria that have successfully incorporated the recombinant plasmid, thanks to the antibiotic resistance gene in the plasmid, will grow. This process effectively isolates our transformed cells, setting the stage for further analysis and protein production. Now let's talk about Step 5, screening of recombinants. To identify the successful clones, we commonly use a method called blue-white screening. This technique relies on the fact that our vector contains part of the LAXI gene, which encodes the enzyme beta-galactosidase. When we perform the screening, we add specific substances like XGAL and IPTG to the growth medium. XGAL is a substrate that turns blue when it's cleaved by beta-galactosidase, and IPTG acts as an inducer, activating the LAXI gene expression. So, if the LAXI gene remains intact, meaning no insert has disrupted it, the colonies will turn blue because beta-galactosidase is active. On the other hand, if the insert successfully disrupts the LAXI gene, the colonies will stay white because the enzyme isn't functional anymore. This helps us distinguish between colonies that likely contain the insert. These are the white colonies, and those that don't, these are the blue colonies. We then select the white colonies for further analysis, as they are the ones most likely to contain our recombinant DNA. This process is a crucial part of TA cloning, as it simplifies identifying the clones that have the desired insert. It's a straightforward, visual way to screen large numbers of colonies efficiently. Let's take a closer look at the mechanism of TA cloning. The first step involves PCR product formation. During PCR, the DNA fragment is amplified, and interestingly, it also receives these three overhangs. These overhangs are crucial because they prepare the PCR products for the next step. Next comes annealing. Here, the A overhangs on the PCR product hybridize with complementary T overhangs on the vector. This hybridization is what allows the insert to pair specifically with the vector, making the cloning process more efficient and straightforward. Finally, we have nick sealing. T4 DNA ligase plays a vital role here by catalyzing the formation of phosphodiester bonds. This seals the nicks in the DNA backbone, resulting in a stable, recombinant DNA molecule. So overall, TA cloning leverages these simple yet effective molecular events: amplification with O overhangs, hybridization with T overhangs, and sealing by ligase to facilitate easy cloning of PCR products. It's a clever system that simplifies what used to be a more complex process. Let's take a closer look at the structure of a typical TA cloning vector. First, you'll notice the origin of replication, or ORI. This is crucial because it allows the plasmid to replicate within bacteria, ensuring we can amplify our DNA efficiently. Next, there's the antibiotic resistance gene, which acts as a selection marker, commonly ampicillin or kanamycin. This helps us identify the bacteria that have successfully taken up the plasmid. Moving on, we see the gene fragment. This is used for blue-white screening, a handy method to distinguish between successful and unsuccessful cloning events. When the gene is intact, colonies turn blue. When disrupted by our insert, they stay white. Then there's the multiple cloning site, or MCS. This is the designated insertion region where we can introduce our DNA of interest. It's a versatile area with multiple restriction sites, making cloning procedures more straightforward. Finally, you'll notice the single T overhangs. These overhangs are essential for TA cloning because they allow for the easy ligation of PCR products that have complementary overhangs. Altogether, these features work in harmony, making TA vectors a powerful tool for cloning. They simplify the process from insert integration to screening, connecting seamlessly with our overall goal of efficient gene cloning and analysis. Let's take a closer look at the advantages of TA cloning. First, one of the biggest benefits is that it requires no restriction enzymes. This means that PCR products can be cloned directly without the need for enzyme digestion, simplifying the process significantly. Second, TA cloning is known for being rapid and simple. Compared to traditional cloning methods, it involves fewer steps, which saves us time and reduces complexity. Third, the high efficiency of this method is a key advantage. The complementary overhangs on the PCR products and vectors significantly increase the chances of successful ligation. Fourth, it's especially suitable for cloning PCR fragments, making it ideal for working with unknown or newly amplified DNA sequences. This flexibility is very useful in many research applications. Lastly, TA cloning involves minimal DNA modification, since we don't need restriction digestion or adding adapters. This keeps the original DNA sequence intact and reduces the chances of introducing unwanted mutations. Overall, these advantages make TA cloning a powerful and convenient tool in molecular biology, fitting seamlessly into our broader workflow for gene cloning and analysis. Let's take a look at some of the limitations of TA cloning. First, it is a non-directional cloning method, which means that the insert can ligate into the vector in either orientation. This can sometimes complicate downstream applications where the orientation matters. Next, there's the issue of background colonies. Sometimes, the vector can self-ligate without an insert, leading to colonies that don't contain the desired DNA fragment. Another important point is that TA cloning depends on Taq polymerase, which adds those A overhangs, but proofreading polymerases don't. This means if you're using high-fidelity enzymes, you might not get those overhangs necessary for TA cloning. And finally, when it comes to inserting large fragments, TA cloning tends to be less stable. It's best suited for fragments less than about 3 kilobases. Larger inserts can be problematic because stability decreases as the insert size increases. Overall, while TA cloning is a useful tool, understanding these limitations helps us choose the right method for our specific needs. Now, let's explore some of the key applications of TA cloning. First, gene cloning is one of the primary uses. It involves inserting PCR products into plasmids, which then can be used for gene expression studies or sequencing. This step is crucial because it allows us to amplify and analyze specific genes of interest. Next, DNA sequencing often relies on cloning PCR products beforehand. By cloning these fragments, we ensure that we have a stable, single source of DNA to sequence accurately, reducing errors and improving the quality of our data. Moving on to mutation analysis, TA cloning is very useful here as well. When we want to study mutated genes, cloning allows us to isolate and analyze individual mutated clones, helping us understand the effects of specific mutations. Another important application is library construction, particularly in preparing cDNA libraries. Cloning cDNA into vectors gives us a comprehensive collection of expressed genes, which can be used for various downstream analyses like gene discovery or functional studies. Finally, recombinant protein expression benefits from TA cloning because cloned genes can be transferred into expression vectors later on. This makes it easier to produce and study proteins in different host systems. Overall, TA cloning is a versatile tool that supports many facets of molecular biology, from gene analysis to protein production, connecting all these applications back to our broader goal of understanding and manipulating genetic material effectively.